from sucrose. But I think it should be pretty close. Okay, so there's that. Then we have, if we come in from mannose, we have a hexokinase that's specific to mannose, and then a different isomerase here. So this is like coming from glucose, comes from mannose, hexokinase, and a different isomerase compared to this one to get you to fructose 6-phosphate. This one is, mobilization of galactose is kind of unusual because it uses these compounds called sugar nucleotides. I don't know if you've encountered sugar nucleotides before. These are the reactions of the fructose. But the pathway here, starting from galactose, is first a galactokinase to make galactose 1-phosphate. And then this funky enzyme here, with a name that I would never ask you to remember, because I could certainly never remember it. UDP glucose galactose 1-phosphate uridyl transferase. The main point being that the cell is using this UDP glucose compound as a way of converting galactose 1-phosphate to glucose 1-phosphate. So GLU1P is made from gal1P. GLU1P can go on through the mutase into the pathway, into the rest of glycolysis. Right? And then this rearrangement here, through this sugar nucleotide, it also involves a couple of epimerases, which are isomerase enzymes that interconvert to chirality at C4 by means of alkylation and then reduction. Okay. The pathway in your book is correct. I keep on forgetting to replace this slide. So this should be NADH goes to NAD, and not the other way around. But in version 6 of the book, it's correct. Okay. Clearly, it can't be right. This is a good exam question. This is getting reduced, and this is also getting reduced. So that doesn't work. So what does this UDP glucose thing look like? I wish you could draw it out so that it makes a little more sense than what's on the slide there. So this is UDP glucose. So here's UDP glucose. This is just a uracil ring up there. And so this is UDP glucose. So UDP linked to the one position of glucose. Okay, and so here we've got galactose 1-phosphate. As the rest of the galactose is on here, up there. So this attacks here. So this goes to glucose 1-phosphate plus UDP galactose. Right, so that enzyme with the long name generates is the catalyst for this reaction. Right. Galactose 1-phosphate here. Here's the attack. Right. So when this leaves, you have glucose 1-phosphate, which is what you want to go through the rest of the pathway, and the UDP. This is UDP now going through U1-phosphate, another one, and galactose is on here. And then the rest of the pathway is just regenerating UDP glucose from UDP galactose by way of those two consecutive redox reactions to flip the chirality at carbon 4, which is the difference between GLU and gal. Questions? So, pretty straightforward. You can just see what some of these other reactions look like. So here we're going from UDP gal, UDP GLU. here. So there's no, once you're using one sugar nucleotide in this, which is here, you're not actually using up a lot of UDP, right? Because it's not in really the substrate of this whole thing. It's gal-1P. The product is glue-1P, and we just have this kind of funky way of making sure that we have a way to replenish the glue-1P as more gal-1P comes through, right? Since we're going through this sugar nucleotide.
So that same UDP is just cycled over and over.
Yeah, so you would have gal-1P, you'd have UDP glucose turning into UDP galactose, while gal-1P would be glue-1P. UDP gal got turned into UDP glue. Gal-1P goes up and it just keeps on going through. So, it's kind of funky. It's like one of these things that cells do. I mean, it doesn't cost a lot of energy except for, the fact that the enzymes have to be synthesized in the cell and they have to kind of be there. And it's a way for the cell to use this other sugar that's coming in. You could easily imagine ways of doing this that had nothing to do with UDP glucose. And so here we have an example of some kind of frozen accident in evolution, whereby this is a very commonly used mechanism in many cells, and it kind of got frozen in this way, not because it's because of physical chemistry or any sort of logical thinking we would think we should do it this way, but simply because it's a historical accident. And biology is full of stuff like this, right? Once it's here, you can sort of perceive that there may be a logic to it, especially if, for example, these sugar nucleotides are used in some other metabolism. Maybe it's coupled in some ways that the amount of the sugar nucleotide has something to do with how much galactose is coming in. So maybe there are bigger picture regulatory questions around it that could be interesting. But as far as just mechanism, who knows? Okay, so next we have fermentation. Okay, so that's the end of the pathways for alternative sugars. So here's the second part of glycolysis, and remember here in step 6 is where we're making an NADH. So since NAD is a substrate, it has to be replenished. And as I mentioned, NADH is typically reoxidized to NAD by some other compound that takes its electrons, which then goes on and passes them on to something else. And so that's how you get your NAD back. But if you have no terminal oxygen, then ultimately you have to get your NAD back some other way. And so that's the fermentation process. Right. So this is the mechanism again. This is the same slide we saw earlier today. The mechanism of the reaction that just illustrates that we need to have another NAD coming back in here, just so that the enzyme can cycle. Right. And so then this is what happens, right? So we have pyruvate at the very end of the pathway, and one way it can go is to get oxidized to acetyl-CoA, but another way it can go is to get reduced to lactate, and in the process of getting reduced to lactate, NADH gets oxidized. Okay. So that NADH didn't do anything with its electrons other than give it to a compound that was right at the end of the same pathway it got made in. And it just got made, 4 steps back in step 6 of glycolysis. Now we're in what we could call step 11 of the anaerobic glycolysis. And in this step 11, NADH that was formed in step 6 is just getting converted back. So this overall process is called a fermentation. Energy is extracted from the sugar. It's stored as ATP. Right? Oxygen is not consumed. There's no change in the concentrations of NAD or NADH because they just got cycled. Right? Also, the redox state of lactate and the redox state of glucose are pretty much the same.